Our last presenter in this session is Suen Lee from the University of Washington, Explainable AI for Health, where we are and how to move forward.
Hi. Is this good enough? Okay, I'm gonna take this out. Okay, hi, my name is Suen Lee. I'm at UW and I'm a computer science professor. Okay, all right. So today I'm gonna talk about some of the research done in my lab on the topic of explainable AI. In a minute, you will know exactly what I mean by explainable AI. It's a subfield of AI whose goal is to understand, interpret the complex machine learning models. Okay, so the work I'm about to talk about is done by amazing students in my lab, in the Ames lab. Students mostly, pursuing computer science degree. And then I have some of the students from the MSTP program, the PhD and, MD concurrent program, pursuing PhD in computer science. I have a couple of them, okay. All right, so we work on a wide spectrum of problems, one in AI and machine learning, pure AI, and machine learning, with a goal to make the AI model more human interpretable. And then you will see how powerful it will be in the biomedical fields in a minute. And then, basic biology, such as, we want to identify cause and treatment of diseases, personalized therapy for, applications in cancer and Alzheimer's disease. We also develop various, clinical AI models, and then also audit them, okay? So you will see some of the work done in my lab on that topic. So this talk will consist of 2 parts. So I have 40 slides, so I will try to go as fast as I can. In the first part, I'm gonna give you many use cases of how explainable AI can be powerful in biomedical fields, with emphasis on biological age estimation, and then also neurodegenerative diseases such as Alzheimer's disease. And then in the second part, I'm gonna show you how explainable AI can do, like beyond, interpreting complex machine learning model, how we can even open, new research fields, such as probably the most relevant topic, which is about, cancer therapy design for precision oncology for this conference, and then also model auditing and then cost-aware clinical AI. So in this talk, you are not going to hear, we are not going to delve into 1 or 2 projects in detail. I'm going to show you many, many examples of how explainable AI techniques can be powerful. And then these techniques are all generalizable to many different problems. So my goal is to show you as many examples as possible so that you can use some of our techniques to your own problem in pharmacogenomics. Okay. All right. So first of all, explainable AI is probably the term I use the most frequently up to now. And what exactly is it? So the goal is to understand the result generated by some AI model, such as imminent dementia. Is there going to be, dementia within the next 3 years for a particular patient? Let's say that kind of prediction was made by machine learning or AI model based on various patient features, such as demographic information, clinical data, sometimes genetic data, and then also cognitive results, and then put them into some black box machine learning model, which is going to generate such a prediction, okay? So it can be pretty accurate. Modern AI models are very good, but it cannot answer very natural questions of why a certain prediction was made. Many of you will think that this is just AI magic, okay? But AI makes mistakes. You will see that in the later part of this presentation. Okay, so to resolve this issue, first of all, why does that happen? It's because with the modern, data sets that has, many samples, you often get to use a complex model rather than simple model. So linear model is one of the simplest models that will give you an idea of which features are the most important. The weights will just tell you which features are the most important, right? But with the complex black box models, such as neural network, deep neural network, then you really can know, the relationship between input and output. So a few years ago, we developed a method that is called SHAP that's widely used in many different fields, highly cited. And then the idea is that you focus on just one prediction, one patient, and then you want to estimate this importance level of each feature specifically for that patient or that prediction. Okay, so you want to identify some individualized factors for the machine learning outcome generated by model. SHAP has a number of theoretical, desirable theoretical properties enough that you can really trust. Probably the most trustworthy XAI method, state of the art XAI method. I'm not going to delve into the all the math behind it, but it has been proven both theoretically and empirically. Okay, all right. So in one of the first applications we applied to some problem of predicting various clinical conditions such as CKD, and then also we applied it to all-cause mortality, and then a problem of predicting surgery duration based on various patients' EHR data, and then we worked closely with the domain experts. For example, for CKD, we worked closely with the UW nephrology to make sure that all the features that are important, individualized risk prediction of a CKD make all sense. Okay, so it's featured on the cover in 2020. It's Nature Machine Intelligence. Okay, all the details are there. Quickly, just switching to some of the one of the newest applications. Well, that was one of the oldest papers from my lab. We also tackled the problem of predicting biological age of individuals. So say that we developed the enable age clock. So the idea is that based on various patient subject information, demographic, your lab test, exam results, and lifestyle questionnaire-based features totaling 825 features, and then we use UK Biobank data to train the model reliably. So the way it works is that, let's say that a patient, a subject, 65-year-old woman, turned out to be 72 in terms of the enable age. And then not only this prediction, you can also see what factors contributed to this biological age, and then the extent in terms of the years. So for example, cancer diagnosis increasing by 1.6 years, and then cystin C, kidney function, increasing about a year. Okay, so when I said desirable theoretical properties, this is one of them. Like this explanation stays sum to the prediction result, and then units are the same. So units of a biological age will be years. Okay, so and then we again worked closely with the experts, domain expert, to make sure that this all factors make all sense. And then the UK Biobank data came with various cause of mortalities, so we can also we demonstrated we can compute biological age and then get this individualized explanations for various causes of mortalities. And then we also performed a genome association study to figure out what potential genetic factors contribute to age acceleration. So in this case, the phenotype will be biological age minus chronological age. Okay, so all results are in the Lancet Health and Longevity. They surprised us by putting featuring our paper on the cover of a December issue in 2023. Okay, so again, we don't delve into too much detail in individual project. My goal is to introduce you a wide variety of applications of XAI methods, so you can find the details in that paper. Okay, so when we talk about cause of mortality, we cannot miss Alzheimer's disease, right? It's so important to identify new therapeutic targets, and then understand the early cellular pathways that goes wrong, okay, before leading to end stage pathologies. So a few years ago, my student Nicosia, who got PhD a few years ago, she collected the data from the ACT study. Kyra mentioned this data, this ACT study based in the Seattle, and also MSCBB from my Mount Sinai, and then RoseMap project. And then at that point, it was all bulk RNA-seq data. There was no, it's not single cell era yet. And then the idea is that we want to use this gene expression levels to predict various measures of Alzheimer's disease severity. So we incorporated a set of, A beta, tau, and the CIRAD score, and so on, and then use a multi-output deep neural network model. It's a complex model. Here, the purpose is not, to predict these neuropathological phenotypes. These are all post-mortem brain, from people who are dead. And then the goal here is to understand the complex interrelations between the gene expression levels and then these neuropathological phenotypes so that we can identify effective therapeutic targets. So the advantages of using this complex model, so deep neural network is one representative class of a complex model, and then the advantage is that you don't need to fully harmonize the phenotype data. So meaning that some of this, ACT, MSVB, or RoseMap data, they can miss some of the AD phenotypes, okay? So the way you train this model, it just allows, still training the model using all samples, okay? And then more importantly, you can capture complex non-linear relationships between gene expression levels and these complex phenotypes. So again, the question of the relationship between gene expression levels and then neuropathological phenotypes will arise, okay? So with this, SHAP method, we can now figure it out, right? I mean, for each of these phenotypes, you will then know, which gene expression levels contributed the most to the high or low pathologies. And then you can also aggregate the result to get a sense of which genes are universally important to explain, this AD overall, AD severity. And then, in our paper, that's the paper in the left, the Nature Communications, we revealed that, some sex-specific, associations between, immune response genes in microglia specifically, and then also AD neuropathologies. Again, detailed biological implications and results are in this paper, the NCOM, Nature Communications paper. Okay, so in the next project, we made more effort to make this AI model, so this genotype, so gene expression to phenotype AI model more interpretable. This can be applied to the case when you use individual genes or some genomic features as the input features of the black box machine learning model. So individuals' genes wouldn't be interpretable as functional units. We all know that, right? Like pathways or biological processes. And then also, unsupervised modeling can help because that way you can incorporate data that do not come with the neuropathological phenotypes necessarily. So in that case, the way it works is that you have input genes. So there's input space, which is 20,000 genes. If you use genetic data, it can be even higher dimensional. And then you reduce the dimensionality into some embedding space. Say, you reduce the dimensionality here, and then so that you can, reconstruct the data well. That's how unsupervised learning works. And then it's going to be deep neural network, so it's going to be a black box model. So what explainable AI means is that, you can pinpoint crucial genes because explainable AI is, it is about identifying important features, okay? So the important genes within that data set. Within the data set, important in, explaining the overall expression variation across the samples within the data set, okay? So we came up with this more biologically interpretable, explainable neural network architecture where we have in this, embedding space, low dimensional space, each latent variable corresponds exactly to a pathway. We use a KEGG or a reactome pathway, and then that is sparsely connected with only the genes that are member. So each latent variable will have a biological meaning. So you can still identify the biological pathways that are important overall across these samples, and then you can also attribute back to the input genes to identify potential driver genes for that pathway. So we applied that to this AD study cohort I mentioned. That way we could incorporate twice more samples because we don't have to worry about neuropathological phenotyping at all. So and then one of the pathways that were important was mitochondrial complex 1, and then we made this hypothesis that it can be a good good mediator for tolerance to A beta toxicity. We did animal-based. So my colleague Matt Kaberlin had an excellent transgenic C. elegans model that basically produces A beta in its muscle. So in a certain age, it's going to show it's going to be paralyzed. Okay, so. And then we show that when you knock down, one of these genes, a dozen genes, and then, it will lead to statistically important increase in this, paralysis, delay in the paralysis time, okay, which indicates that, it can lead to some promising pharmacological avenue. So we noticed that, capsaicin is the drugs that actually, suppress this complex. So after this project, I decided to eat more peppers. Okay, so remember 40 slides. Okay, so in the next project, we switched our attention to the single cell data. So more and more, we saw these Alzheimer's disease studies. They produced the data from single cell transcriptomics data from this the postmortem brains, and then we noticed that in single cell data they often do contrastive ask a contrastive scientific question in terms of what is the difference between unhealthy tissue and then healthy tissue, and then treatment or exposure to drug or exposure to pathogen, and then also CRISPR perturb the genome. What would be the difference between them? We are not interested in the aspects that are common. So we came up with explainable biologically interpretable architecture, where in the embedding space you separate the latent variables into shared variables, literally it's shared by background and target cells, and then also salient variables. You just turn this the background cells salient variables to be activation 0. Okay, so when you do that, when you apply to cancer cells treated with a certain drug, and then use untreated cells as a background, and then you will see cells behave differently in the salient space. It's only because this nuisance cell type specific signal. Those of you who have handled single cell transcriptomics data, so cell type specific signal is a biologically dominant signals. Okay, so it's going to be all absorbed by this shared variable. These are not the aspects you are interested in if you want to identify some way to personalize the medication. In this case, it would be TP53. Okay, so in our the last year Nature Methods paper, we developed a method that can be applied to many single cell data sets for this kind of a contrastive analysis. Now we are applying that to Alzheimer's disease to understand what is the expression signatures that are specific to Alzheimer's pathogenesis, regardless of cell types of tissues, and then also the aging as well. Okay, all right. So in the second part of the talk, I'm gonna just talk about what we can do beyond just explaining the model. Okay. And first example is about cancer combination therapy, which was done by Joe Janizek. This is one of the students from the MSTP program. He just got the PhD and MD and then got matched to Stanford. So we focus on this combination therapy because, as we know, cancer therapies are increasingly becoming combination and it has great efficacy. And then, we focused on AML, acute myeloid leukemia, in this study. But, then choosing the optimal combinations will be hard. There are already many, individual FDA-approved anti-cancer drugs, and then there are tens of thousands of pairs of drugs, and then, patient characteristics, genomic, cancer characteristics will be important. So, explanations in this application will be incredibly important. So we developed this express method that takes gene expression levels of blood, and you pick, a certain pairs of drugs, and then use drug-specific features such as molecular structure and gene target. The drug is known to target, and then you use a black box machine learning model that performs really well in predicting the synergy between these two drugs you are considering. Okay, you can test many different drugs this way, and then not only that, it also gives you explanations in terms of which genes were important for synergy or the other way, not synergy. And then that way you can also perform pathway-based analysis like GC, get a sense of which pathways were important for that particular decision. Okay, so to just one example, we considered all this, gene level importance levels for all combinations that contain venetoclax that, that targets a specific gene BCL2, and then it was one of the genes that had, high importance SHAP values in this, in terms of this gene level importance. Okay, so and then we focused on which genes are universally important across many drug combinations and many different patients to get a sense of if there is underlying principles in terms of what's important for the synergy between any two anti-cancer drugs, and we identified these two genes that are well known to be associated with poor, the prognosis, and then the genes that are known to be, associated the level of differentiation of cancer. So some people call this like stemness of cancer, and then we did a further analysis and then made a conclusion that the genes that are universally important in predicting synergy, drug synergy, are involved in hematopoietic cell lineage. So, and then the level of differentiation. So in our paper, it's this Nature Biomedical Engineering last year, published last year, we did many analysis and XAI-based analysis, and then also biological analysis to make a conclusion that stemness, level of differentiation, can be considered as axes to design combination therapies. And then, if you use two drugs, if one drug is known to work well for the less differentiated cancer, you better use the other drug to, known to work well for the better, more differentiated cancers. Okay, so again, the details are in this Nature BME paper 2023. So quickly, introducing the next use case of explainable AI, it's about AI auditing. So in 2020, during the pandemic, there was a surge of AI models that takes X-ray data and then predict whether that patient has COVID-19 or not. We learned that many of them learned shortcuts rather than genuine pathology. It's actually a very scary result. So when you test, the prediction performance within one data set, even in cross-validation setting, it appeared to perform really well. But if you take, some data that's completely external to that hospital, then it's not going to work well as much. Okay, so, and then we did XAI. So XAI is about what? It's about, identifying important features. Okay, so in this case, you have X-ray data. Pixels are features there. Okay, so you can see then, using the most basic XAI analysis, you will see which regions in the XAI X-ray image was considered important by that machine learning or AI model. Okay, so for example, in this case, the X-ray data, COVID negative, and then it's right that the model used the clear lung basis as a basis for predicting COVID-19 status, okay? But it's not quite right, the laterally markers like this. You don't want AI model to rely on that kind of signal or patient medical devices the patient was wearing, right? You clearly don't want your AI model to rely on that kind of signals that are irrelevant to pathology. So we showed many kinds of analysis and then basically stopped the field from moving in the wrong direction. There were already 6 published papers and hundreds of related models out there that were going to be used and then learned the shortcut. Okay, so next year, Nature, this featured this paper and then emphasized the importance of actually breaking, into the black box of the AI model. And then they put this really, really nice photo of the two first authors. These are my 2 students from the MSTP program. Again, this is Joe, who's going to Stanford, and then this is Alex. He just finishes his PhD, computer science PhD, and then went back to the MD program. Very busy now. Anyway, so now our next target for AI auditing is dermatology. So you can find very easily in your cell phone app store, that takes your skin image and it's going to tell you whether that's a melanoma or not. We took 5 models, 2 academic models, 1 Nature paper, and then 2 commercial devices. We perfectly replicated all these systems, and then 1 competition winner, number 1. And then, we ran into some interesting technical challenges. This is what, the example where, the XAI itself, computer science itself, it should also improve. So let's take a look at this saliency map. Is it useful? So this is the homoscopic image. You can also use a cell phone image for these systems. And then it's going to just, just pointing to the entire lesion in this image. It's not interpretable at all, right? So you need to basically do more things. So we developed, based on generative AI technology, developed a new XAI technique in collaboration with Stanford dermatology, so Dr. Roxana Denshu at Stanford. And then the idea is that you want to make slight modification to your original image such that it just crosses the classification boundary by that, this AI model you are auditing. So that way you can understand what this AI model was thinking. So in this case, you suddenly see when this image becomes a malignant, the minimal change was this region, bluish, region. It's a blue-white veil that my collaborators said, and then I learned a lot of dermatology terms during the project. And then you can then, after that, you can do systematical characterization in terms of what features were perceived by each AI model you were auditing. And then and then the result is basically can look something like this. So these are the names of the systems we audited. And then for each of these systems, you will have many pairs of this image that has a modified image that's normal, and then benign, and then malignant counterpart. So by looking at this differences between them and identifying major visual features, you can know how each visual feature is perceived by each model. Okay, so for example, darker pigmentation, it was completely differently perceived, completely differently by one of the systems, right? And it's not consistent with the literature. So that way you will know when this model was failing, you will know why that happened. Okay. All right, so this is just one example of how generative AI can be useful. I wrote a short perspective article that shows you, some broad promises of such counterfactual AI model. You can find it February 2024 Lancet. And then we also, worked on, so some slide was missing. One slide is missing. Okay, sorry. So the idea is that the second way, so we worked on completely independent method that also makes this kind of a model more interpretable. So when you see a image like this, do you think of this as a bunch of pixels? No, humans don't think that way. Humans think about concept. What is the border type? What is the size of the lesion? What is the color? Is it multicoloration or darker pigmentation? People will think about, you don't have to think about even dermatologists. I mean, humans think of this kind of image as a set of concepts, right? So we developed a method that uses an image text foundation model, and by fine-tuning the OpenAI CLIP model with 100,000 image and text captions from the textbooks and then PubMed articles, okay? So the idea is that model like this, the goal is to bring image and text into one embedding space. Remember, embedding means low-dimensional representation, okay? So, and then that way, when you have just image, and then for a set of the terms, that makes sense, dermatology terms that, that are semantically meaningful, then you can basically featureize. You can generate another set of features for each image in terms of, what aspect is represented how much, okay? So this paper is published in Nature Medicine a few months ago. Okay. So now just, last example, very quickly, it was done by Gabe, who was a year ago, he got matched to Harvard Medical School. And then, it's emergency medicine, which was his PhD, project that was published in Nature Biomedical Engineering in 2022. So the motivation is that, a lot of healthcare AI models, people think about just accuracy. But in a situation like, ER, say you have a trauma patient in front of you bleeding, and then these ER doctors want to predict whether this patient is going to have acute traumatic coagulopathy, which is a very dangerous bleeding condition. You require, massive transfusion. So accurate prediction is important, but also you wanna diagnose fast. Okay, and some features that require deeper look and more time, you don't want to fully rely on these features if you think about the time cost. Okay, so we worked with UW Medicine, Nathan White, and then and used the trauma registry data that has 46 features, and then we worked with many medical directors to get a sense of how much time it takes to get each of these features. And then what XAI basically does is to estimate the importance numerically, quantify the importance of each feature, and then we combine that with the feature cost, in this case time, and then we did a joint optimization to come up with accurate but also fast model. So the result will be something like this. It's not going to be standard ROC curve. Y-axis will show area under the ROC curve accuracy, prediction performance, and the x-axis will be model cost. How many minutes in total it takes. And then compared to co-AI, our method, PEC score is an existing clinical score, which takes 8 minutes to gather all features. We take, with co-AI, you can use a model that uses, fast. You can gather your feature much more fast. Okay. As I said, all the methods I'm presenting today are generally applicable, broadly applicable. We applied the co-AI scheme to the problem of predicting mortality in ICU patients, where there are many a lot more, already clinical scores, especially QSOFA is designed for exactly that purpose. You want to be efficient in terms of the feature gathering. Sorry, okay, yeah, co-AI was, but I don't know how to go back. Okay, so quick summary. I showed you how explainable AI can be impactful, and then, one aspect I didn't really delve into is that this is incredibly hard computer science problem, and that we still, you may not recognize this, conferences, top conferences in AI, NeurIPS or ICML, JMLR. These are all top machine learning venues. We actively publish in that venue to accurately estimate the features importance. When you have many features or correlated features, there are all sorts of difficult scenarios. And then we apply to wide variety of application, spanning the medicine and then cancer biology, precision medicine, Alzheimer's disease, where there's no drug. I mean, no long-term effective drugs yet, and then you wanna understand the biology of it. Okay, so what I talked about is only a small portion of our research. You can find more about our research, and then as you might have noticed, we work on many different things. So I had to label every single paper in our publication list in terms of A, B, and C. You probably would be most interested in the C category, and or some in B. A is very mathematical. So if you have insomnia, and if you want to fall asleep, just go to my homepage, and then just pick any paper in the A category. Okay, so again, I just want to emphasize that all the work, all the plots, and I haven't generated a single plot I showed you today or in our publication. I haven't written a single line of the course. It was all done by my PhD students, and then we were generously funded by NIH, NSF, American Cancer Society, and Chan Zuckerberg Initiative. I made this size of the logo roughly proportional to the size of the font. Thank you.
Questions? We have some time. The other speakers want to come up for additional questions? Or maybe we lost Kira. So questions for,
yeah.
Go ahead.
Yeah.
Thank you for your excellent talk. I just curious, like in the literature, quite often I see machine learning.
Yeah.
Some is called decision tree. Some of them called AI. I just wondering.
I know. Yeah, these terms.
Explain the difference.
Explaining the difference. So AI is a bigger field. I mean, if you want to be really precise, I mean, these days, AI, machine learning, they are like used interchangeably. I think if you want to be really precise, AI is a bigger field that includes machine learning. So machine learning is specifically about models. You have a statistical model. You train the model to fit your training data. And then, it can be used for robotics or computer vision and many other fields. So combining, machine learning field, because the theoretical aspect is really important, right? So that's why there is, this field of machine learning. And then, computer vision, robotics, and then, computational biology also, and then computational medicine, clinical AI, clinical informatics, these are all, combined is collectively called AI. Yeah, I think that would be the most, precise. But it really doesn't matter, these days when you say AI. I like the term AI more because I want to be more inclusive. But then when you have to refer to a model, particular model, then I tend to, say it's a machine learning model. Then, it's about really the statistical model itself. Yeah. Oh.
I'm just curious, what is the regulatory impact? Like, is this a medical device? How does this kind of thing get looked at by the FDA?
Yeah. So I had to remove the slide to make it a 30-minute talk. It used to be like 45-minute talk. Yeah. Well, FDA has to use this, right? I mean, so this see this is dermatology AI example, right? So at the end of 2022, GigWire. It's a Seattle-based, the tech-oriented media. And then they asked me what to expect in 2023 in digital health. So I didn't hesitate. I just put my, in that slide I removed, I put the article, and then I said, in 2023, which is already past, right? 2023, we will see these AI devices, health devices that come with XAI functionality. And FDA is going to use XAI for, approval process. Didn't happen. So I didn't, prediction was not correct, but I was going to say we are moving in the right direction. And then sometime last year, I got invited to give a talk, interest them with the idea of XAI, and then, this awareness. I mean, what we see from this X-ray project, COVID-19 project, and then also, dermatology project. So I think at the end, we have to incorporate this, SHAP or other, XAI functionality, both in, this AI devices or, and this FDA approved, but not happened yet. But I think we are moving in that direction.
All right, thank you.